sun on our trip today in early November.
Today we're at Pandapa's Pond in Virginia, meeting field zoologist Dr. Derek Hennen, whose work focuses on the taxonomy and systematics of Appalachian myriapods. Arthropods are in the subphylum Arthropoda. A variety of subphylums fall under this, including the subphylum Myriapoda, containing arthropods such as millipedes and centipedes. Centipedes have a leg per segment and are carnivores. Most are harmless to humans, and their venom is primarily fatal for other insects and animals. The millipede, however, has 2 legs per segment, eating decomposing plants, and if threatened, will simply coil up, sometimes secreting an irritating fluid. And interestingly enough, both are egg layers.
Fashion accessory. This is a tool called an aspirator. So it is a rubber hose connected to a little collecting vial, and we have a small little filter there. And so this is so that I can suck up smaller bugs, whether it's insects, millipedes, centipedes. And so it's important because for some of these species that I'm after, they're very fast and they're also very small. So you don't necessarily want to grab them. You might squish them. They might drop some of the legs that you need them to have so you can identify the species. And so by doing this, I can just suck them up and they'll fall in here and be all nice. They don't think they're being, grabbed or anything, so they're not dropping any legs, they're not getting damaged, and it's just a quick little... And so, it's just a nice little tool that I started using after a lot of frustrations of getting damaged specimens. They don't have a waxy outer layer on their exoskeleton like a lot of insects do, and so they lose water really easily. And so they're always trying to avoid desiccation, drying out, because then they just, they die after that. They're too parched. And so they're always looking for somewhere that's protected, under stones, under logs, under bark sometimes. So dryness is really their main enemy in life. So intermittent stream, it does have some water, but they tend to be sort of at the bottom of some of these ridges. And then it gets it nice and, it's nice and shady, it's very damp. You see all the, it's not only rhododendron leaves, it's stuff from up higher. We got oaks and pines and beech and stuff. But generally it's pretty good millipede habitat. The rhododendron, you can kind of see up there, they can sometimes form these very interlocking, sometimes they're called rhododendron hills. And they're just hard to get through. Like sometimes you have to go back if you get stuck in one and can't get through.
Derek explained to me the tedious method of raking done for his fieldwork, just simply kneeling down and going through leaf litter patches near trees, under rocks, or under logs, not knowing if you'll find anything for hours in one area or the next. After a bit of raking, we decided to move on.
Writing the locality labels and everything. Because I've dealt with many labels from old collectors who they put down everything. Especially in Blacksburg, and underneath it runs the other way. So if you want to stand in front. No, we're probably not going to see many snakes today. So there are a couple of species that are really only found under dead logs and wood and stuff. Oh yeah, right there. Yeah, so these are 2 stone centipedes. It's an order called the Lithobiomorpha. They're also found under logs and leaf litter or whatever else, it's just a nice little common name.
What are you putting them in?
So this is rubbing alcohol,
Okay.
so about 70% ethanol.
Nice.
Yeah. So that preserves them. With things like insects, you can just pin them because they're very hard-bodied, but Millipedes and centipedes, they need to be preserved in alcohol because they're soft-bodied. But even the millipedes that are hard-bodied, if you just pin them, they just kind of disintegrate because the essentially the body matter between the rings kind of dehydrates and breaks apart, and then all the rings kind of fall. So they look much better when you've got alcohol. You can work with them scientifically. But you never know what's under the log till you overturn it.
Hopefully it disappeared.
There's a little millipede actually. Right here. Oh. The millipedes are sometimes harder to suck up. So this is the genus Cladogona. I call these the sausage millipedes because their order name in Latin is Cordumatida, and in Latin, corduma means sausage. And they're kind of sausage-shaped, I guess, if you kind of squint. And this group of millipedes, they actually they have spinnerets at the end of their body, so they can actually spin a silk-like protein, kind of like a caterpillar. So they make little cocoons to molt in. You don't really want standing water. And then if you see that, you just kind of like go up the hill slowly a little bit. Oh, here we go. That's a twisted claw millipede. So we don't really want to eat these millipedes because it'll be a really bad time. You could die. Like some of the larger cherry millipedes that are like an inch and a half long, they've got enough cyanide inside their bodies to kill about 18 pigeon-sized birds.
Wow,
Yeah.
crazy.
This one doesn't have quite that much, but it can still pack a punch. And this might actually be one of the new species that we described a few years ago.
Really?
Because we collected them around here.
Can you tell if it's a male or a female?
I think this one is a female. That's kind of based on, number one, how many of these things I've seen, but also the size of the legs. They're kind of thin. The males, their legs tend to be a little bit thicker. And the males are sometimes also a little bit smaller in body size than this.
Oh, the males are small.
Yeah. Yeah, the females are generally a little bit larger. Mostly that's to accommodate eggs, because they just store their eggs inside their body cavity. And millipedes are kind of interesting because they're progoniate, so all their genitalia is actually closer to their head than at the end of their body, like we're used to with a lot of other things. This might be. So millipedes, when they molt, all arthropods, they need to molt their exoskeleton. And so they need to find a secluded place to do that, because that's when they're most vulnerable, because they're very soft when they first molt, so they're easy pickings for a predator. So, different groups, they do different things. The flatback millipedes, they will actually form this little molting chamber out of dirt and feces. So I think that's what this is here. So who knows, maybe it was hers. But, it's almost like a little urn or something, and they're right there in the center. And the reason that they also include poop in their chamber, well, never mind, that's actually for their egg chambers. But, the poop kind of helps bind the dirt together. And so you'll sometimes find these little empty chambers, you're like, what are these things? And it's like, oh, it's a millipede molting chamber. Sometimes you can actually see the imprint of their butt where they were kind of pushing everything together. I think that's what you got right there in the end.
So funny.
Yeah.
I don't know what it is.
Oh, it's a millipede. Oh, nice, yeah. This is the violet-ridged millipede. This is Cambala annulata. And this is in a group that a lot of its diversity is in the tropical region. Here in the eastern U.S., in this entire order, we've only got about maybe a dozen species or so. So there's all these ridges along the body there. And these are kind of interesting. Probably won't be able to see it without a major close-up, but they do have eyes, but they're just in a line kind of down the side of their head.
Whoa.
Whereas usually with millipedes that have eyes, they're in kind of a triangular arrangement. And these have chemical defenses. These are the ones that'll stain your hand kind of a yellow color, and then it'll kind of fade to an iodine bruise. So if I kind of push her enough, she might do that. They'll kind of curl up as a defense. You'll see that.
Yeah.
Mostly she's focused on just getting away.
Yeah.
So these ones you sometimes find in a big aggregated group, especially when it's been pretty dry. They'll kind of all huddle together, I guess. But again, right behind the head, that first column that first segment is enlarged, called the column, and that's what they're using to kind of bulldoze stuff. They'll kind of put their head down and just push to get through places. And these often get into caves as well.
I think it's funny when they accidentally flip.
Yeah. So this is in a group sometimes called the bark centipedes or the tropical centipedes. If you've ever seen like these nature documentaries where it's in a tropical place, they're waiting outside a cave and grab a bat that's flying out and eat them. So this group can get up to like a foot long, but the species we have down here, they're just going to be a couple inches at most. Oh, hey, here's a soil centipede. It's a species called Strigamia bidens, and it's always this really nice scarlet red color. And if we stretch that out, that's probably about 2-3 inches long. And they can have a lot of legs, anywhere from like around 57 to like 83 or so. And so you can see, it looks very worm-like.
Yes.
And how it moves, it's kind of sending the head out, but if I touch it, it kind of like just retracts. It's kind of anchoring the end of its body there, and it'll just kind of go away. And these are, they're predators of little soft-bodied soil arthropods. These are so small you don't have to worry about it biting you. The actual forcipules on this one are relatively puny. It's a little bit awkward when it's on the surface. Really, it's going to be down underneath the soil, and all those legs can hold on to soil particles and just wind itself around however it needs to. And so with centipedes, their forcipules, the venom jaws that people are always scared of, those are actually a pair of modified legs. So their actual like mouth parts, the mandibles and everything else, they're tiny, they're kind of hidden below those big forcipules.
After a bit more of searching around the forest, raking through leaf piles, and a little help from a surprise visitor, we had found it. The rare species Derek had told us about beforehand. One of the hardest millipedes to spot.
So what's the name of it again?
Biotis carolinus. So this is one of those little cards for Emerald Ash Borer ID. I probably had it for like 10 years. So with a lot of these particularly rare species, you're looking at some pretty small things. Like all the big ones are easy to find, they're easy to kind of learn about. You really have to be digging to get the small stuff.
After a lot of celebrating, Derek decided there was one more thing he needed to show us before he wrapped up for the day.
So for this, we have this nice little contraption here. So this is a leaf litter sifter or leaf litter concentrator. So we've got our two handles here on the inside. We've got a nice little mesh grid, and so you're picking up all these leaves here, sticking them in there, and then you're just kind of churning it up, and it falls into this nice little handy dandy cinched bag. Then after that, you've got, instead of just getting a bunch of leaves that aren't going to have a bunch of stuff, you want all the smaller stuff below it that you can really concentrate, and then you put that into a contraption called a Berlese funnel, which essentially you spread the litter out also on a metal grid, put a light bulb above it, and then all the critters that are in the leaf litter then falls down they go down through the dirt, fall through that mesh grid, and then they go down into a cup of alcohol that you have there, and then you can look at it later, and then you see all the little stuff that you never see just by eye in the forest. You just shake that around. It's kind of nice and get a good churn going in it. Just a little vortex of leaves. Also helps if there's like salamanders or snakes there, they don't come with you. There was, for about a year, I was coming every 2 or 3 weeks to take leaf litter samples and trying to figure out what species of millipedes and centipedes are here. And so I did that for a long time during all different seasons, and I would get different species during each season. And at the end of that, I came out with about 60 species of millipedes and centipedes. So here in the leaf litter, we just have the young, the juveniles of all the beetles, flies, other bugs that just make the forest really work. And so without this nice protected layer of leaf litter and all the habitat that it covers and all the protection that it gives to those young, you don't have any of the processes going on in the forest that you really need for it to function.
After many successful finds, we decided to wrap up for the day and head home. I feel like I learned about a lot. The experience opened our eyes to what we might walk over every day, what millipede or centipede could be under that log or in that pile of leaves by that tree. And it was really great to see and learn about the life of such an underrated arthropod. They deserve more recognition, and hopefully in the future people will give them a little more credit for what they do, because they do a lot of work in their own hidden world under us.